Bismillahirrahmanirrahim, and welcome back to this next video. And this is another video in the series of videos on the insulin signaling pathway. In the last video, we discussed about the insulin receptor. That insulin receptor is a tetrameric structure. It contains two alpha subunit, it contains two beta subunit, and the tyrosine kinase activity that lies in the beta subunit. Then I've told you about some of the important information about the receptor, that the gene is located on the chromosome number 19. It have got 22 exons and the beta subunit that actually contain the tyrosine kinase activity. That is what we discussed in the last video. Now in this particular video, I want to focus on the signal in this particular insulin signaling pathway, which would be the insulin. I've told you in the last video that whenever you talk about a cell signaling pathway, the two important things are the receptors and the signal. We have already seen the receptor in the previous video, and in this particular video, I'll be focusing on the signal, which is the insulin. If you talk about the insulin, the mature insulin, by that I mean that those particular insulins which are able to interact, or their particular form of the insulin which is able to interact with the insulin receptor. So that particular insulin is made up of two chains. One is known as the A chain, which is made up of 21 amino acid, and the B chain, which is made up of the 30 amino acid. So the mature insulin, which is actually the signal, this is 51 amino acid long. Now if you've got two chains, there should be something responsible for the stability, for the structural stability, that can maintain the A and the B chain together with each other. Now this particular strength is provided by the disulfide bonds, and the disulfide bonds, they are of, you can say, two types, I would say. One, they are the interchain disulfide bond, in which the amino acid number 6, which is actually a cysteine, so let me refer to that as the A6. So this would be one cysteine in the chain A, that is going to make a disulfide bond with another cysteine, which is present at position number 11. So this is an intra-chain disulfide bond, and this is responsible for the structural stability. Secondly, there are interchain disulfide bonds. What I mean by that is that there are disulfide bond between the chain A and the chain B. Now, at which particular position these disulfide bonds are present? At position number 7 of the chain A, there is a cysteine amino acid. At position number 7 of the B chain, there is a cysteine amino acid, and they are going to make an interchain disulfide bond. Another interchain disulfide bond that is made between the amino acid number 20, which is actually a cysteine, of course, and the cysteine, which is present at position number 19 in the B chain. They are going to make these disulfide bonds, and the collective power of these disulfide bonds, the interchain and the intrachain disulfide bond, that is responsible for the stability of the signaling molecule. Now the question is that, is insulin directly produced in this particular form? And the simple answer is no, because the insulin that is produced in an immature form, which need processing, and that particular immature form of the insulin, that is known as the preproinsulin. What I mean by that is that there are additional amino acids present in the insulin, which must be chopped off before the insulin it can get into an active form where it can interact with its receptor. So let us have a look at the preproinsulin, how the preproinsulin looks like. If you look at the preproinsulin, you're actually talking about 110 amino acid long protein. In this 110 amino acid long protein, you are going to see four parts. One that is represented in green over here, that is known as the signal peptide. The one that is represented in red, that is the B chain, and of course the B chain is going to be present in the mature insulin. This blue one is the A chain, and again, this is going to be present in the mature insulin. And then you have got the C peptide. So in the mature form, you are not going to see the signal peptide and the C peptide, and the A and the B chain, they are going to be seen in the mature form. So when the signal peptide, the B chain, the A chain, and the C peptide, all of them, they are present, this particular insulin is known as the preproinsulin. So why the signal peptide and the C peptide, they are present? So let us have a look at the function of the signal peptide and the C peptides. Now these signal peptide, they are very important for the targeting of the preproinsulin to the endoplasmic reticulum. So because of the signal peptide, the preproinsulin that is going to move towards the endoplasmic reticulum after it has been synthesized by the ribosome for modification. Now, this signal peptide, when they make the preproinsulin to the ER, we are no more interested in the signal peptide because that has performed its function. The function of the signal peptide was to get this preproinsulin into the endoplasmic reticulum for processing. Now, once the preproinsulin that has reached the endoplasmic reticulum, we are going to cleave the signal peptide because it has performed its function, and this cleavage is carried out by an enzyme which is known as the signal peptidase enzyme. So when the signal peptidase enzyme, it cleaves the signal peptide, the preproinsulin, it gets converted into the proinsulin. So we have modified the preproinsulin by the removal of the signal peptide, thereby converting that into the proinsulin. Now, the proinsulin, again, it has to go for further processing to become the mature insulin, like the folding and the disulfide bond formation. So this is going to be the proinsulin. This one is the B chain, this one is the A chain, and this one is the C peptide, as we have already seen in the last image. Now, when the signal peptide that has been removed, the proinsulin has now been converted into the proinsulin. Now, what is the function of the C peptide? Now, the C peptide, that's very important for the proper folding and the stabilization of the insulin molecule. And once that particular folding and stabilization has been achieved, we are going to get rid of this C peptide. So, how to remove the C peptide to convert the proinsulin into the mature insulin? This function is performed by an endopeptidase, and what the endopeptidase do, it is going to remove the C peptide, so only the B and the A chain, they are going to be there, and they are going to be proper folded, and they are going to be properly stabilized to give you the mature insulin. So this is what you are going to get at the end of the functions of the signal peptidase enzyme and the endopeptidases. You are going to get the A chain, you are going to get the B chain attached to each other by the intrachain disulfide bond and the interchain disulfide bond to give you the mature insulin, which has the ability to bind to the insulin receptor. So this is how the signal in the insulin signaling pathway looks like. Now, in my next video, I'll be focusing on the pathway. We have already discussed the receptor. Now we have discussed the signal. So in the next video, I'll be focusing that when the signal and the receptor, they interact with each other, how that is going to lead to the insulin signaling pathway.